Hello everyone, and welcome to another recorded lecture of BIO1 online. Today we'll be discussing Chapter 9, which is on meiosis and sexual reproduction. So, reproduction doesn't have to be sexual. Reproduction can be asexual or sexual. And asexual reproduction is done by several types of organisms that include bacteria, archaea, and some single-celled eukaryotes, such as this amoeba right here. And in asexual reproduction, there's one parent that replicates its DNA and splits into two cells. This is basically mitosis and cytokinesis. You're starting off with one cell, it duplicates its DNA, and divides into two. So asexual reproduction gives us genetically identical offspring. Right? So this amoeba this is the parent amoeba. Each of these daughter amoeba is exactly the same. They're genetically identical to this parent. So asexual reproduction is basically just like mitosis and cytokinesis. You're cloning cells. In sexual reproduction, the DNA of the offspring comes from two different parents. So sexual reproduction, in contrast to asexual reproduction, you have two parents. And in sexual reproduction, the male parent contributes sperm cells, and one of which will fertilize a female's egg cell to become the next generation. And in sexual reproduction, we generate genetically diverse offspring, right? So this is the key point to sexual reproduction. We're getting genetically diverse offspring. But for sexual reproduction, we need to attract a mate and produce sex cells, which requires a lot of energy. And energy is costly to the organism. So what's the benefit of sexual reproduction? And why is sexual reproduction so common? Well, we said that with sexual reproduction, we get genetic diversity among offspring. And genetic diversity is worth the energy because diversity increases the chance that some offspring will survive in changing environments. So as we know, before I even go to this example, Earth is always changing. Temperatures are changing. There are different parasites that come about. There are different chemicals in the atmosphere. So if all organisms were the same, right, and there weren't a lot of diversity, that doesn't give nature a lot to choose from, right? If there's a huge change in the environment, that means all of the organisms will be affected the same way. So suppose in a genetically identical population, everyone's doing great. They're making mass productions of each other. They're just mass cloning, all the same. But let's say there's an ecological disaster. Suppose there is an ice age, or there is a volcano, or something, some parasite emerges that is able to compromise a whole population of organisms. That's it. That's the end to that population. If there's no diversity, they're all the same. So if one of the organisms will be affected by this disaster, the entire population will be wiped out by this disaster. In a genetically diverse population, so you have offshoots, right? You have some branch points where you have different combinations of genes coming together. So you have diverse genes. So yes, an ecological disaster will get rid of a lot of your organisms. But because there is some mixing and matching of traits, right? Because there is genetic diversity, you have some likelihood that some offspring from these original parents will survive because they have different combinations of advantages. They have different adaptations. So this is why diversity increases the chance that some offspring will survive when the environment changes. And again, environments always change. So sexual reproduction is very beneficial. So to summarize, genetic variability will increase the chance that at least some individuals will have a combination of traits that allows them to survive and reproduce. Right? Even if there are going to be some poorly suited individuals over here that are going to die, you still have some survivors that will be able to then pass on those genes and keep the population going. So before we discuss sexual reproduction further, let's talk about what types of cells you're made out of. So somatic cells are body cells, we said, and most of your cells are somatic cells. And we said somatic cells are diploid. They have two copies of each gene. One gene came from mom, the other gene came from dad. The other types of cells we have are gametes. Females have eggs, males have sperm, and gametes are not diploid. Gametes are haploid, meaning they have half the number of chromosomes that body cells have. And remember, that's important because we want two haploid gametes to come together to form the first diploid cell of the next generation. Right? If sperm and egg both had two sets of chromosomes and they came together, then the next generation would have four sets of chromosomes. So we want sperm and egg to have half the amount of chromosomes that body cells have. So when they come together, they'll form a diploid organism. Gametes are located in the gonads, which are ovaries in females and testes in males. And gametes have the DNA that can be passed on to offspring. So it's only the DNA in sperm and egg that get passed to the next generation. Within every cell of our body, so of all of our somatic cells, remember those are diploid, that means we have 23 pairs of chromosomes. So if we were to look at all of our chromosomes, chromosome pairs 1 through 22 don't influence sex at all. And we just call those autosomes. So we have 23 pairs of chromosomes as humans. Chromosome pairs 1 through 22 we call autosomes. And again, those are just chromosomes that are the same for both sexes. The sex chromosomes, X and Y, determine sex in mammals. So that will determine whether the individual is male or female in mammals. Females have 2 X chromosomes, whereas males have 1 X chromosome and a Y chromosome. What you're looking at here is called the karyotype, which is the entire set of a person's chromosomes laid out. And again, chromosomes only look like this when they're in prophase or metaphase. So these are fully condensed chromosomes. They don't usually look like this. So this karyotype shown is taken from what? So you can pause here and tell me what you think. So since there is an X and a Y chromosome, we say that this is a biological male. Right? It's X and Y chromosome. This karyotype shown is taken from a gamete in G1, a somatic cell in G1, a gamete in G2, or a somatic cell in G2. This is a little tricky, so please pause here. So the first thing to recognize, let's see, is it a gamete or a somatic cell? So since we had 2 copies of every chromosome, we know this can't be a gamete. This has to be a somatic cell. We have 1 copy from mom, 1 copy from dad. The question now is it in G1 or G2? And if you recall from the cell cycle, that G1 is before DNA replication and G2 is after DNA replication. So does this represent a cell that is regular, like just a normal cell that's hanging out, or is this a cell that's ready to divide? And the answer is this is a cell in G1. This has the normal set of chromosomes, right? We would have 46 chromosomes, or 23 pairs. If this were in G2, we would have double the amount of information, double the amount of chromosomes, because then that would have to split into 2 cells. So the answer is B. So whereas sex chromosomes determine whether an individual is female or male, chromosomes do not determine gender. And I just want to talk very, very briefly about the distinction between sex and gender. Sex refers to the biological and physiological characteristics that define males and females. And this, like sex, is determined by chromosomes and hormones, which is ultimately produced by genes. Genes control hormones. And again, nothing is strictly genetic, right? Everything has an environmental influence. So it's not just nature or nurture. All physical characteristics are shaped by our environment. And in biology, there's a very simple definition of male and female. In biology, the sex that makes the larger of the 2 gametes is called the female. So since an egg is a lot larger than a sperm, the egg-producing individuals are considered female in biology. The sperm-producing individuals are called males. The same thing is in the plant kingdom as well, right? So the larger of the 2 gametes The ovule is female, where the pollen, the smaller of the two gametes, is male. So in biology, the sex that makes the larger gamete is called the female. That's sex. Gender is out of the realm of biology. That's more of a sociology topic. Gender refers to the socially constructed roles, behaviors, and attributes that a given society considers appropriate for each sex. So gender might be influenced by chromosomes and hormones, but it's not determined by them. And gender is specific to a time and place. So what is masculine in one era might be very feminine in another, right? It's all arbitrary. And again, gender is up here, right? Not down there. So there's, again, an important distinction between sex and gender. We're not going to talk about gender in this course in biology. We'll talk about sex, which talks about the difference in making gametes. Very briefly, I want you to actually just look at these myths based on this book I really like called Evolution's Rainbow. So true or false, an organism is solely male or female for life. That is false. Many plants and many animals are both male and female in a lifetime, and some can actually switch. Males are bigger than females on average. That is also false, right? Especially in fish, the females are larger. Females, not males, give birth. True or false? This one's false, right? In many species, such as the seahorse, the females deposit eggs in the pouch of a male, and the male will give birth. Males have XY chromosomes and females have XX. Well, that's false. The reverse is true in birds. Some species don't even have sex chromosomes, and sex is determined by temperature. Only two sexes occur in nature? Nope, that is very false. Some species have 3 or more sexes or can be intersex, having both male and female reproductive parts. Males have penises and females lactate. Believe it or not, that is also false. So the female spotted hyena has a penis, and the male fruit bat can lactate. So there are always exceptions. And males are dominant and control females. That could not be farther from the truth. In fact, in many species, females control males and do not like the dominant males. So it's good to just get an orientation of reality as it is in the animal kingdom and not in our human societies. So back to biology. Let's do a quick refresher about chromosomes. We said chromosomes are made of chromatin. Chromatin equals DNA wrapped around histones. Each chromosome is one long DNA double helix wrapped around histones, condensed and wrapped tightly and tightly and tightly until it looks like one half. This is a duplicated chromosome. But one DNA double helix gets wrapped around histones until it gets more and more compact. In picture 2, what is DNA being wrapped around? I just said this. The answer is B, histones. What is labeled red in these pictures 3 through 5? So pause here. The answer is B, these are centromeres. What phase of the cell cycle occurred between pictures 3 and 4? So please pause here. The answer is C, S phase, right? Here we had one DNA molecule, and then in S phase, DNA polymerase copies the entire thing, so we have 2 copies. Chromosomes would appear as in picture 5 during which phase of the cell cycle? And the answer is D, during prophase, where the chromosomes condense. And then in metaphase, they're going to meet in the middle of the cell. And by anaphase, these 2 halves will be pulled apart from each other. They won't be together. So this brings us to a very important discussion that must be understood very well. So humans have 2 copies of each chromosome, right? We said we have 23 pairs of chromosomes. 1 copy of each came from mom. The other copy of each came from dad. There's a word called homologous. That means having the same structure. And that's a very important term to understand. So suppose we have chromosomes 1 through 23 from mom, and we have another copy of chromosomes 1 through 23 from dad. We could say chromosome 15 from mom is homologous to chromosome 15 from dad. What that means is both chromosomes 15 have the same sequence of genes and the same order. They basically have the same identity. They're the same locations of every gene in the same locations. So homologous means they have similar structures, but it doesn't mean that they're identical. All right, so this could be chromosome 15 from mom, and then on top there's the gene for eye color, then there's the gene for enzyme A, and then there's a gene for cytochrome C. But are homologous chromosomes identical? The answer is no. For example, chromosome 15 from mom always will have, or all chromosome 15 in humans, I should say, will always have the eye color gene in the same location, enzyme A gene, and the cytochrome C gene, right? All chromosome 15s are homologous to each other because they match up, but they're not identical. Because suppose dad might have green eyes, a green eye color gene, and mom might have a brown eye color gene. So homologous pairs of chromosomes have the same types of genes in the same locations, but there might be different forms of that gene. We call different forms of genes alleles. So there is an eye color gene, but there can be a brown-eyed allele or a green-eyed allele. Right? An allele is like a flavor of a gene, right? So the eye color gene can have different flavors. It can have different alleles. So to summarize, homologous pairs of chromosomes have the same types of genes in the same locations, but there might be different versions of each gene at each location. So again, this might not make perfect sense. You might have to review this many, many times before it clicks, but it must click. So what happens to these chromosomes in S phase of interphase? So normally we have 23 different chromosomes from mom, 23 different chromosomes from dad. And in S phase of interphase, chromosomes are duplicated. Now we have two identical copies of each chromosome. This is where it gets a little tricky. So now we're going to have a duplicated chromosome 15 from mom and a duplicated chromosome 15 from dad. Each copy of a duplicated chromosome is called a sister chromatid. So these two are sister chromatids, and these two are sister chromatids. Sister chromatids are identical, right? The sister chromatid was made after DNA polymerase just copied this chromosome. So it's important to know the difference, right? So during S phase, chromosomes are replicated by DNA polymerase. And a duplicated chromosome consists of two identical sister chromatids. So here we have a homologous pair of chromosomes because they have the same structure, same order of genes. We have one from mom and one from dad. During S phase, each of these were copied, and now they have a sister. So this represents one duplicated chromosome. A duplicated chromosome consists of two sister chromatids. So I hope that makes makes some sense. And we have access to gene maps of many species that show the relative positions of genes on a chromosome. So for example, in the fruit fly, Drosophila melanogaster, we know what exact genes are on chromosome 2. So on chromosome 2, we know that the antenna gene is first, then this eye gene is, then the wing gene. So this is called the gene map. Right, so on the human X chromosome, we could map out all of these different diseases. So we know which disease alleles are located where, because we have a gene map. So again, homologous chromosomes will have the same gene locations, but they might have different alleles. So let's try to see if this question makes sense. When during the cell cycle will chromosomes look like the picture below? So pause here, and you have to use process of elimination to narrow this down. So this is how chromosomes would appear in interphase, before S phase. So in a normal cell, we would have one copy of chromosome 11 from mom, one copy of chromosome 11 from dad, one copy of chromosome 17 from mom, one copy of chromosome 17 from dad, dot, dot, dot. We would have this from chromosome 1 through 22. And then the X and or Y chromosome. So we're only going to see this before S phase. After the chromosomes get replicated, we're going to see double this. Each of these will have a sister. So if I just wanted to, right, so after S phase, that's going to have a sister, right? And then this is going to have a sister. Right? And then these two will be identical to each other. Right now, this chromosome over here is not necessarily identical to this chromosome over here. They are homologous. They're not identical. So pause here, looking at these, this picture over here, and tell me which statement is true. So the answer to this question is C. 1 is identical to 2. These are sister chromatids. But 2 is homologous to 3. Right? Chromosome 2 is homologous to chromosome 3. So let me test you again on this concept. How many copies of chromosome 10 do you have in your cells during G1 of interphase? So normally, how many copies of chromosome 10 do you have in your somatic cells? The answer is 2. You have 1 copy from mom, 1 copy of chromosome 10 from mom, and 1 copy of chromosome 10 from dad. Those 2 copies are homologous to each other. What is the difference? Well, I just said this answer, right? What is the difference between the 2 copies of chromosome 10 and interphase? Right? The answer is 1 copy of chromosome 10 is from mom, the other is from dad. Right? So they're homologous. In G2, how many copies of chromosome 10 will you have? So after the DNA is copied, that means you're going to have 4 copies of chromosome 10. Right? Because your copy from mom got duplicated. That means 2. And then your copy from dad got duplicated. That's another 2. So you're going to have 4 copies of chromosome 10 after DNA is copied. And then, of course, once the cell divides, you're going to end up with 2 copies of each again. So that makes sense. So we said body cells are diploid. So how many chromosomes do human muscle cells have? Right. So the average, let's say an average, if it's a single-celled muscle cell, you would have 23 pairs of homologous chromosomes. Right, 46 total. And diploid cells, right, means you have 2 copies of every chromosome. So 1 copy of each chromosome comes from each parent. How many chromosomes do human sperm cells have? We have 23 single chromosomes that are haploid, right? Haploid cells only have 1 copy of each chromosome. Body cells, like a hair cell or an eye cell, have 2 copies of every chromosome. And there are some muscle cells that have multiple nuclei, so that's an exception, but I'm just trying to simplify it here. So diploid cells have 46 chromosomes. Haploid cells have 23. So when a haploid egg cell and a haploid sperm cell come together during fertilization, we get the first diploid cell called the zygote, basically a fertilized egg. And the zygote is the first diploid cell of the next generation. That diploid cell will continue to do mitosis throughout development until it becomes an adult, and only during puberty will meiosis start in these adult individuals to produce more sperm and egg. So now we're going to talk about mitosis and how it compares to meiosis. We spoke about mitosis last time. We said it keeps the number of chromosomes constant. So if you have a 46 chromosome cell and that undergoes mitosis, you're going to get 2 cells with 46 chromosomes each. And mitosis happens throughout the body and throughout the lifespan of the organism. Meiosis is different. So we're going to talk about meiosis now. And meiosis divides chromosome number in half. In meiosis, what we're going to see is haploid gametes will be made from special diploid germ cells. A germ cell is basically a sex stem cell. And these stem cells can only produce gametes. They're only located in the gonads. So these are destined to just become sperm in a male or egg in a female. So again, in meiosis, the goal is we're making haploid gametes. This only occurs in the gonads, testes or ovaries. And again, we're getting genetically unique haploid cells. So we're not doing identical copies like in mitosis. We're mixing and matching genes in meiosis. So we're getting genetically unique haploid cells in meiosis. And again, only certain cells do meiosis that are found in the gonads. These are special germ cells that are destined to become sperm and egg. So only certain cells in the gonads do meiosis. And it's meiosis that will produce these genetically unique cells that allow for the diversity from sexual reproduction. So meiosis is key to sexual reproduction so we can get that diversity, right? So we can withstand any possible changes in the environment, and our species has a better chance of surviving and reproducing. And unlike mitosis, meiosis can only be completed after puberty. So if a zygote of a ferret contains 40 chromosomes, how many chromosomes are in a ferret's sperm cell? So pause here. And the answer is B, right? 20. So there must be 20 in a sperm cell, 20 chromosomes in the egg cell. So when they come together, that zygote had 40 chromosomes. What is the chromosome number of a human sperm cell not in meiosis? So an average human sperm cell has how many chromosomes? The answer is B, 23 single chromosomes. Right? So meiosis, unlike mitosis, consists of 2 cell divisions. So, and again, only certain types of cells can do meiosis to begin with. So these are special diploid germ cells in the gonads, go through 2 cell divisions to make these unique haploid cells. We call these 2 divisions meiosis I and meiosis II. And meiosis has the same steps as mitosis. So meiosis will start with interphase. And then we have meiosis I, which consists of prophase I, metaphase I, anaphase I, and telophase I. We don't use prometaphase as a note. So meiosis I, we have PMAT, prophase, metaphase, anaphase, telophase, and then cytokinesis. Then we have 1 cell division. And then there's actually a second brief interphase without any DNA duplication. So now we're just dividing these in half. So meiosis II consists of prophase II, metaphase II, anaphase II, telophase II, and a final cytokinesis. The key point is that there's only one time where DNA is replicated. So once the cell divides, they're not going to duplicate DNA again. So therefore, we're going to get half the amount of genetic information that we started with after meiosis II is complete. So let's just go through an overview of what we're going to see in meiosis I and II. First, the diploid cell must undergo interphase, where the cell is going to grow, produce proteins, get ready, make sure the environment is right. So the DNA will be replicated, and each of the 46 chromosomes will consist of 2 sister chromatids. So we technically have 92 sister chromatids, 92 chromatids total. In meiosis I, again, so bear with me here, we're going to see the 23 pairs of duplicated homologous chromosomes find each other. So chromosome 1 from mom will find chromosome 1 from dad. They're going to line up and exchange genes. So each chromosome will physically align with its homolog, and the homologous pairs will then exchange genes and then separate into 2 separate cells. So that's what happens in meiosis I. In meiosis II, it's very simple. It's basically just like mitosis. We're going to have these 2 sister chromatids separate. Each cell divides, and now we're going to have 2 separate sister chromatids in meiosis II. right? And at the end of meiosis II, we're going to have 4 haploid nuclei with unduplicated chromosomes. If you're scratching your head, it's understandable, right? This is not very easy. It takes multiple reviews of meiosis to make it make sense. There's a really good animation through Amoeba Sisters on demystifying chromosome numbers in meiosis, and that's in the learning map. I highly suggest reviewing that so you understand what we're talking about when we're saying chromosomes are being divided and going from diploid to haploid. So now we can discuss how genetic diversity is achieved in meiosis, right? What's the whole point of sexual reproduction? Diversity. So how is that diversity achieved? It's during crossing over and recombination. So during meiosis I, specifically we'll see this happens in Prophase I, 2 duplicated homologous chromosomes pair up and will exchange pieces, literally physically exchanging genes. And this will scramble the genetic material, mixing and matching different alleles. So let's go back. So originally we only have one of each. In a regular non-dividing cell, we have one of these from mom. This is one chromosome, or sorry, we'll say from dad, and this from mom. And then what happens in interphase of meiosis I is each of these get replicated. So now we have each chromosome has a sister. So this is right after interphase of meiosis I. Now in prophase, we have this really interesting step where the homologous chromosomes find each other. So chromosome 1 from mom found chromosome 1 from dad, and they're physically exchanging pieces. They cross over at chiasmata. A chiasma is singular, chiasmata are plural, so they cross over in multiple locations along the chromosome. And where they cross over, they recombine their genetic information. So what started off, we had a diploid cell. We had one copy from dad, one copy from mom. We ended up just mixing and matching genes, and then the cell divides. So each of these cells is now haploid because it doesn't have 2 separate copies, right? This has 2 separate copies. This is diploid. We still have 2 separate copies. This is diploid. But now we only have 1 copy. Granted, they're mixed and matched, so they're a little different, but we call this a haploid. This is now haploid. This is now haploid. And in meiosis II, that haploid DNA will then divide into 2 separate cells. So after meiosis II, each of these sister chromatids will separate from each other. And we're going to end up with 4 haploid gametes. Some of these gametes will be just like one of the parental combinations. So some of the gametes, like some of the sperm and egg, will have exactly the same chromosome intact just from the one like dad had. But some of these gametes will be recombinant, meaning they'll have some alleles from mom and some alleles from dad. So 50% roughly will be recombinant, mixed and matched, and 50% will be just like the parental. Let's try to understand what this means. Suppose dad has blonde hair and blue eyes, and mom has brown hair and brown eyes. What that means is this cell can have blonde hair and blue eyes, right? So that means the organism that develops from the sperm will have brown hair and blue eyes, blonde hair and blue eyes. But it's also possible that the blonde hair gene mixed with the brown eye gene. So it's possible to have now blonde hair and brown eyes because mom and dad's genes mixed and matched. Right? This is giving us more possibilities. So again, this is illustrating the importance of sexual reproduction. We're mixing and matching genes, so we're getting different possibilities, different variations. So maybe one of these 4 will be more likely to survive. And again, in this case of hair color, it's not going to make a huge advantage. But you can suppose that maybe this is a disease. The little a means disease, if you have the little a. So anything with this little a in it will be a diseased allele, where anything with the big A will be healthy. So now you're basically giving your offspring a chance to get the healthy gene and not just the unhealthy gene. So this all occurs during Prophase I of meiosis. Both replicated homologous chromosomes pair up in what we call a tetrad. Tetra for 4. So we have the 4 chromatids lining up, and they do a little handshake. They physically cross over and they exchange genes. And those that exchange genes we call recombinant chromatids. So they line up and they mix and match their genes to create genetic diversity. So let's watch this video together so we have a better understanding of what's going on in crossing over.
Crossing over during meiosis allows for recombination of genes between homologous chromosomes. This alters the arrangement of genes on the same chromosome. Crossing over occurs frequently during Prophase I of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent, and the other member came from the other parent. In this example, one parental chromosome had alleles uppercase A and uppercase B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of genes is not altered in this case. Now consider the case where there is crossing over in the region between the 2 genes. Since the crossing over is only between 2 individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over, and will still have the parental combination of alleles, either uppercase A and uppercase B, or lowercase a and lowercase b. The other 2 will be recombinant, with either uppercase A and lowercase b, or lowercase a and uppercase b alleles. Thus, crossing over shuffles the alleles and brings about genetic recombination in the gametes.
So now let's go through meiosis I step by step. In meiosis I, we're going to see that homologous chromosomes pair up, cross over, and then separate. So in interphase, the DNA replicates. It's the only time in meiosis where we have DNA replication is before meiosis I. And then in prophase, we have chromosomes condense and become visible. The nuclear envelope breaks down, and the spindle fibers begin to form from the centrosomes. Right? This looks just like prophase of mitosis so far. But something unique happens during Prophase I of meiosis. Right? Crossing over occurs. And that happens when all of the homologous chromosomes pair up. Right? All the homologous chromosomes line up next to each other. And then they shuffle their alleles like we just saw. So they're doing this crossing over here in Prophase I, mixing and matching genes. Now in metaphase, we have some chromatids are parental, look just like the parent, the original, but some are recombinant. Some have part mom's genes and part dad's genes. And in metaphase, the paired homologous chromosomes will line up along the center of the cell. Right? This is important. Each member of a homologous pair attaches to a spindle fiber stretching to one pole. So what we're going to do is separate homologous chromosomes. So this pair, I'm going to use my little pen here. This pair is linked to one centromere. This pair is linked to the other one. So these are going to be separated, right? Again, we're separating the entire pairs. So these 2 guys will go to one cell, and these 2 pairs will go to the other cell. Right? So it's different than in mitosis here. And the way that homologous chromosomes line up can be very different. So this is what gives us what's called independent assortment. So chromosome pairs aligned randomly during Metaphase I, and that again gives us even more combinations of chromosomes, even more genetic diversity. So there are different possibilities. At Metaphase I, the pairs of homologous chromosomes line up at the cell center. Suppose we could have one alternative where all of the blue chromosomes are on top and all of the red homologs are on the bottom. So if that were to happen, then we can have this arrangement of gametes after meiosis. Right? We'll have cells that have only the blue and only the red. But suppose there's a different alternative. Suppose that chromosomes 1 and 2 had blue on top, but just for this third pair of chromosomes, these two were switched. So now when they split up, we're going to have a different combination of moms and dads chromatids. So there could be a third alternative or a fourth alternative. So in this example, we have just 3 pairs of chromosomes. And if a germ cell has 3 pairs of chromosomes, that means there are 4 possible arrangements during metaphase. Right? You can have all blue on top, all one on top and all the other. You can have 2 and 1, right? The 1 and the 2. So any of these combinations can arise during Metaphase I. And with 3 pairs of chromosomes, that means we have 4 possible arrangements that can give us 8 possible gametes, right? 2 to the third. Since we have 3 pairs of chromosomes, each of those 3 pairs of chromosomes has 2 possibilities. So that gives us 8 possible gametes. In humans, how many possible chromosomal arrangements can we get in Metaphase I? So we have 23 pairs of chromosomes, right? So with 23 pairs of chromosomes, that means each gamete contains one of over 8 million possible chromosomal combinations. So again, that's 8 million possibilities for different combinations in just one of the sexes. Right? In fertilization, we're getting males and females both contributing their chromosomes. So we have to take that number of 8 million and square it. So there's actually, so there's 8 million possibilities in eggs that could combine with any of the 8 million possible sperms of the partner. With that, we get over 70 trillion different possibilities for zygotes. That's without crossing over. That's if all the chromatids remained intact. Even if crossing over occurred once, we can have this many genetically different zygotes available. Right. So that's even, we square this again. So this is how no two people are alike, right? This explains how no two individuals are alike, because crossing over occurs many times in each chromosome. So this gives us trillions and hundreds of trillions of possible combinations of zygotes. So again, in Prophase I, we had crossing over, and in Metaphase II, we had independent assortment, right? Depending on where those chromosomes aligned, they're going to be separated in different combinations. Right? That's what we saw here. So crossing over and independent assortment contribute to genetic diversity and meiosis. Now we're up to Anaphase I. And Anaphase I is when homologous chromosomes separate to opposite poles of the cell. This is important. In Anaphase I, homologous chromosomes separate. Write that down. All right, homologous chromosomes separate in Anaphase I. The sister chromatids remain intact, joined at the centromere. So these get pulled apart. And then finally, in telophase and cytokinesis, we have the opposite of prophase. So the nuclear envelope will reform around the chromosomes. Chromosomes may temporarily decondense, the spindle disappears, and cytokinesis divides the cell into 2. So that is the end of meiosis I. So let's watch what happened so far.
During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but two events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase I concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase I, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase I, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In Anaphase I, the homologous chromosomes separate and move toward opposite poles. Meiosis I ends with Telophase I, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow.
So a second interphase precedes meiosis II in many species, but the DNA does not replicate a second time. That is critical. And meiosis II looks just like mitosis. So we're going to start with what we call prophase II. Spindles form, nuclear envelopes break down, the chromosomes are condensed. In metaphase II, the chromosomes will line up along the equator of the cell. In Anaphase II, sister chromatids separate to opposite poles of the cell. That is key, right? In Anaphase I, homologous pairs separated. In Anaphase II, sister chromatids separate. That is key. In Telophase II, the nuclear envelopes will form around the two separate sets of chromosomes. And cytokinesis will then divide the cells, and at the end, we're going to get 4 non-identical haploid daughter cells. All right, so overall, we just divided up genetic material after mixing and matching. So now's a good time to watch this video and the ebook that goes through each of the stages of meiosis. Meiosis I, and then meiosis II. And again, it's a very visual process, so it pays to watch this many, many times. All right, and here again is an overview. We said interphase was the one time DNA was copied, and this is a cell with that's 2N. Right, this is a diploid cell. So we have one of each. Then in interphase, each of these was copied. In prophase I, those copies formed a tetrad. They lined up and they crossed over. Crossing over and recombination. The chromosomes lined up at the equator, and the homologous chromosomes were pulled apart. The cells were split into 2, and right away they go into prophase II, where now they just meet in the middle, and each of the sister chromatids are pulled apart. So after cytokinesis, we get 4 haploid cells, some of which are recombinant, because they went through crossing over and recombination. Some will be parental. And remember, This just shows one chromosome, but in our human cells we'll have 23 of these. Again, I'd really want you to understand how meiosis is different from mitosis. Meiosis had 2 cell divisions, while mitosis only had 1. In mitosis, we never see homologous chromosomes pair up, right? Chromosome 1 from mom and chromosome 2, or chromosome 1 from dad, never see each other in mitosis, right? They both just get duplicated and they both separate. Meiosis gives us haploid cells that are not identical. Mitosis results in diploid cells that are identical. And I'd like you to pause here and look at this table that compares meiosis 1 to mitosis. This compares meiosis 2 to mitosis, which is very similar. And this is a summary of everything. So it's a good slide to kind of just refresh and make sure you understand how mitosis compares with meiosis. And this is a good image that breaks down some of the differences too. So in meiosis, you can see there's crossing over. Here, there's no crossing over in mitosis. This is very important to understand. In Metaphase I, the homologous chromosomes align next to each other. This is chromosome 1 from dad. This is chromosome 1 from mom. This is chromosome 2 from dad or mom, and this is right. They're finding each other. Whereas in mitosis, this can be chromosome 1 from mom, and that's chromosome 2 from mom. Right? They don't line up. So chromosome 1 from dad is not trying to find chromosome 1 from mom. Right? They're just all lining up on top of each other. They never mix and match genes. Only in Metaphase I does chromosome 1 from mom meet chromosome 1 from dad, and do they exchange genes. Right? So this could be chromosome 1 from mom. This could be chromosome 1 from dad. This could be chromosome 2 from dad. This could be chromosome 2 from mom. Right? They all align at the metaphase plate on top of each other. And then in anaphase, the sister chromatids separate. In anaphase I of meiosis, the homologous pairs separate. Right? So those are very important distinctions. So now it's a good time to watch the video in the ebook that goes through the comparison of meiosis and mitosis. So now we'll start with part 2. And in part 2, we'll see how meiosis leads to the production of sperm and egg in humans, but other organisms produce spores. So I just want to talk about this in one slide. So fungi, some protists, and certain plants produce haploid spores. And these spores are produced by what's called the sporophyte generation. So there's a certain form of plant called the sporophyte, which is the diploid sporophyte generation, that produces haploid spores by meiosis. So this fern, for example, produces spores by meiosis. Those spores are haploid. Those haploid spores grow into what are called gametophytes. And the male and female gametophyte are haploid. And these gametophytes, as they're named gametophyte, they can produce the gametes. And those gametes can then fuse together the same way as sperm and egg would to create a diploid zygote. That zygote will then grow into that mature diploid sporophyte. Again, that sporophyte will do meiosis to make spores. The spores will grow up into a gametophyte, which is also haploid. And the gametophyte makes gametes. Right? Those gametes then come together to form a zygote, and that will become the sporophyte again. This is called alternation of generations. Right? So plant life cycles include this alternation of generations between multicellular haploid and diploid individuals. Right? And just compare that to the human life cycle, where we don't have alternation of generations, right? We are always diploid, and we produce haploid gametes. Those come together to form another diploid organism. So we're always diploid, we just produce haploid gametes. So spermatogenesis is sperm production in males. So meiosis in male mammals is part of spermatogenesis, and males do not produce sperm until they reach puberty. And while females produce only one egg around each month, males produce millions of sperm almost every day. So spermatogenesis will give us 4 haploid sperm cells for each germ cell that undergoes meiosis. So within the testes, we have what are called spermatogonia. These are diploid germ cells that can either become another spermatogonia or can become what's called a primary spermatocyte. The primary spermatocyte is what can undergo meiosis I. So the primary spermatocyte is a diploid cell. Crossing over will occur. And in meiosis I, we get 2 haploid secondary spermatocytes. In meiosis II, we know that sister chromatids separate. So in meiosis II, we're going to end up with 4 haploid spermatids. And those then mature, they form a flagella, they do all these different modifications to become a mature sperm. Right? So we said secondary spermatocytes complete meiosis II. We get 4 spermatids, each of which will specialize into a mature sperm that kind of looks like a tadpole. And oogenesis is egg production in females. So meiosis in females is part of oogenesis. And the production of egg cells, called oocytes, is complete before the female's birth. So even before birth, these oogonia, these germ cells in the ovaries, produce primary oocytes, but these arrest in prophase I. So these primary oocytes are in the ovaries, but they arrest at prophase I until puberty. Then a hormone called FSH, follicle stimulating hormone, stimulates a potential egg to complete meiosis I around every 4 weeks. So starting from puberty, and every 28 days, a hormone is released into the bloodstream that tells one of these primary oocytes to start meiosis again. And that meiosis will continue until Metaphase II. It then waits this is the ovulation period. So once the hormone is released, FSH, and meiosis is continued to Metaphase II, that oocyte is basically waiting for a sperm. Only if a sperm enters will meiosis II be completed, and will the egg be able to be fertilized. What's interesting is that after we start off with one oocyte, and we only get one egg at the end of oogenesis. This is different than in spermatogenesis. So let's review why it's different. We said oogonia are in the ovaries. They can become another oogonia or become a primary oocyte. These primary oocytes are what undergo meiosis I. This happens before birth. Once puberty starts, all right, meiosis I will continue from prophase until meiosis II. Meiosis II will only be completed if fertilization occurs. And what happens is, after the first meiosis, only one of the 2 cells gets a lot of cytoplasm. This one gets less cytoplasm. So only this secondary oocyte, not this one. This is called a polar body. This is a secondary oocyte. So they're divided unequally. And only this secondary oocyte will be able to become the fertilized or the mature egg. So the secondary oocyte will undergo meiosis II. Again, not dividing symmetrically. It's asymmetric division. So we're getting one nice big egg called an ovum. And then we end up getting 3 polar bodies that are discarded. So once again, we started off with a primary oocyte. After meiosis I, we got a secondary oocyte and a polar body. That polar body gets discarded. The secondary oocyte will undergo meiosis II, where there's another asymmetric division. So we get one ovum that has a lot of the cytoplasm, all the organelles and the proteins and everything needed, and then that polar body gets discarded. And it's this ovum that again gets fertilized by the sperm. So this is a good slide to pause on to make sure you understand spermatogenesis and oogenesis. So it is possible to have two individuals that are genetically identical. And we see that in identical twins. We call identical twins monozygotic twins. Monozygotic twins. And that's because they come from one zygote. So one sperm cell fertilized one egg cell and formed one zygote. But instead of that zygote just dividing and becoming one organism, that zygote split into two and became two separate embryos. So each embryo then developed independently, and these are clones of each other. Right? This has the combination of DNA from sperm and egg. These two embryos will have the same combinations of sperm and egg. Right? So monozygotic twins come from one zygote. Again, monozygotic twins still have differences. Right? We spoke about that with epigenetics. So clearly, there are other factors other than genes that influence different physical characteristics. The other types of twins are called fraternal or dizygotic twins. And in this case, two eggs happen to be fertilized by two separate sperms. So instead of one egg being ready to be fertilized, two eggs are ready to be fertilized. And two separate sperm cells fertilize two separate eggs. And that makes two separate embryos that are no more related than any brother and sister that are born 10 years apart. So two zygotes develop as two separate individuals. So again, they only share 50% of the genetic information. So finally, I'd like to talk about some human genetic syndromes. And oftentimes, changes in chromosome number and structure can cause various syndromes. You can do this nowadays with a lot of new technologies, but in the past, we had to use what's called the karyotype, remember? So you would actually take some of the fetal cells, and you can stain the chromosomes and lay them all out, and you could count and make sure that there's the right number and structure of chromosomes in the fetus. And again, now we can sequence DNA, and it's very minimally invasive. We can also understand genetic inheritance a lot better, so we can look at pedigrees. But overall, we want to have the right number and the right structure of chromosomes, and that's called euploidy. Euploidy is when you have the proper number of chromosomes. So that's what we hope for. But an error in meiosis can give us what's called aneuploidy, which is an incorrect number of chromosomes. It could be an extra chromosome or a missing chromosome. And this is caused by non-disjunction during meiosis. So errors in chromosome number are caused by non-disjunction. So normally we have a pair of homologous chromosomes that separate in meiosis I. But suppose that both pairs of chromosomes are brought to one cell and the other cell has nothing. That's a non-disjunction in meiosis I. Right? You can also have a non-disjunction in meiosis II. Right? Normally, we'd expect this cell to divide into two sister chromatids. But if this doesn't disjunct, right, this doesn't separate, we're going to get one cell with two sister chromatids and one cell with none. If we continue, right, with just this one non-disjunction, you're going to end up getting two cells with two copies of each chromatid and then two cells with nothing. So if a normal healthy sperm were to fertilize each of these possible eggs, we're going to only get these two that are normal, right? Because we want to have one copy of every chromosome from mom, one copy of every chromosome from dad. Here, we're going to have two copies of moms. Here, we're going to have no copies of moms, right? Here, we're going to have two copies of mom, just like over here, right? And here we have no copies of mom's DNA. So non-disjunction gives us either an extra copy of chromosomes or a missing copy of chromosomes, right? So we can say normally we want 2n, we want one copy of each. But it's possible through non-disjunction to get 2n plus 1, we got an extra copy of a chromosome, or 2n minus 1, we're missing a copy of a chromosome. So this is called non-disjunction. It's when the chromosomes fail to separate. And again, this meiosis 1 would be a failure to separate at Anaphase I. Right? This is where Anaphase I, we usually separate these two. And then meiosis 2 is a failure of Anaphase II. Right? Because in Anaphase II, we separate this into two separate chromatids. So this is what we want to have, ideally. And again, this is what we'd expect. At Metaphase I, the homologous pairs line up, and in normal meiosis and Metaphase II, and Anaphase II will have 4 separate haploid gametes. If there's a first division non-disjunction, that means in Anaphase I, both pairs of homologous chromosomes will go to one cell, the other will have none. That will give us gametes that are missing a chromosome, and some gametes that have an extra chromosome. Suppose meiosis 1 goes just fine, and this is what should happen in Anaphase II, but suppose that there's a non-disjunction in meiosis 2, and both sister chromatids end up in one gamete. That means we're going to have an extra chromosome. So as a result of non-disjunction, it is possible for a sperm or egg cell to have an extra copy of chromosome 21. And what that can mean is that when sperm and egg come together, the zygote will have 3 copies of the 21st chromosome. So that's called trisomy, tri for 3, trisomy 21. So if you have an extra copy of trisomy 21, that is the most common cause of Down syndrome. An affected person with Down syndrome has distinctive facial features. Intelligence varies greatly. Some children have mental impairments, whereas others learn very well. Many people with Down syndrome are able to live very fulfilling, productive lives. There was a good show a few years ago called Born This Way on A&E that talks about these different individuals with Down syndrome and how they face their challenges and lead lives just like us. But again, this extra chromosome presents some issues. And people with Down syndrome also have an above average risk for leukemia and Alzheimer's disease. And that probability of giving birth to a child with trisomy 21 increases as a woman ages. And I'll show you a graph of that in a couple of slides from now. Because for a woman of 48, the incidence jumps from 1 to 10. So it gets 10% chance if a woman is 48 years old compared to 1 in 3,000 at age 30. So there's a 10% chance of having a child with Down syndrome if a woman is 48 years old, compared to, again, 1 in 3,000 if a woman is younger than 30. You could also have changes in sex chromosome number. So you could have many copies of an X or a Y chromosome, or too few, and that can also lead to abnormalities. Turner syndrome. Turner syndrome is an example of a disorder that affects 1 in 2,000 females, where you're missing an X or a Y chromosome. You only get one X chromosome. So Turner syndrome is XO. They're still considered females because they have an X chromosome. And we only consider individuals with a Y chromosome male in biology. And some symptoms of Turner syndrome include short stature, sexual underdevelopment, some heart defects, and some learning disabilities. But that, again, varies by the individual. But again, this is having only one copy of an X chromosome instead of 2. So looking at these 2 karyotypes, we can see some abnormalities. Over here, we'd normally expect either an X or a Y, but now there is a missing X chromosome, and that's what gives us Turner syndrome. Here, there is an extra X chromosome, and that is Klinefelter's syndrome, and that affects about 1 in 750 males. So there's different disorders that result in non-disjunction of sex chromosomes. You should be familiar with Turner syndrome, which is XO, and Klinefelter's, which is XXY. There's also triplo X syndrome, where you have 3 copies of the X chromosome, and there's Jacob's syndrome, which is XYY, where you get 2 copies of Y chromosome. So we said you could have an extra copy of an entire chromosome, but you could also have errors within chromosomes. And mutations in chromosome structure can have major effects on an organism. These types of effects can happen from radiation exposure to errors that are from crossing over. So normally, we'd want to have, let's say, genes A, B, C, D, E, F, G, and I shouldn't say genes. These are like regions. So normally we'd have regions A through G on our chromosome. But it's possible that part of the chromosome will be deleted. Again, this can be because of radiation exposure, some kind of error in crossing over, or something else. But this is a deletion. We're missing a whole piece of a chromosome, and that could be thousands and thousands of genes. It's possible to get a duplication, where we get, instead of A, B, C, D, E, F, G, we get D, E, F, D, E, F, G. So we're getting extra copies of genes. It's possible that a portion of the chromosome will be inverted. So it'll go A, B, C, F, E, D, G. That could be harmless, or it can be very harmful, right? Depending on just on which genes are located here. It's possible that all the genes will still be able to be expressed, even if they're not in the same order. But maybe the gene expression might be altered. It's also possible to have what's called a translocation, where separate portions of different chromosomes cross over. Right, so normally we'd want these genes on this chromosome and these genes on a separate chromosome. But in a translocation, 2 parts of chromosomes switch. So it's crossing over between non-homologous chromosomes. So we do not want that to happen. So here's a summary of these different types of alterations in chromosome structure. And again, these can be harmless, or they can lead to a miscarriage, right? So oftentimes, if a gamete has extra genes or missing genes, there's just a miscarriage. So most of the time, these fetuses will not be born if they have crazy, abnormal chromosome structures. So an example of a deletion that you can survive is in Williams syndrome. So Williams syndrome is a loss of the end of chromosome 7, which contains the gene for elastin. And without the elastin gene, it gives these individuals heart and cardiovascular problems, distinctive facial features, but also some learning and intellectual disabilities. So again, you can still miss a part of a chromosome and still survive, but you're not going to be average, so to speak. You could have a translocation that can result in Alagille syndrome. So chromosomes 2 and 20 switch places in people with Alagille syndrome. And that leads to these are clubbed fingers, but also heart defects and liver disease. So translocations often break genes. And sometimes that can cause cancer. So that's not good. Other times it's harmless. So this is an interesting chart that shows a range of different chromosomal abnormalities and how many fetuses per 100,000 pregnancies are either aborted spontaneously because of these abnormalities, or how many are able to be born. And you can see just from looking at this, trisomy 21 is one of the few instances where you can have a live born individual with a trisomy. Most of the times, if you have a trisomy of any chromosome, right, you see a huge portion of spontaneous abortions. So it's a very rare chance, right? If you have only individuals with trisomies of 13, 18, or 21 have a chance of even surviving. And out of 100,000 pregnancies, about 8,000 of them had major chromosomal abnormalities. 7,500 out of the 8,000 with chromosomal abnormalities were not born alive. So again, it's very rare. It's rare enough to have a chromosomal abnormality, right? Only 8,000 out of 100,000. But if you do have a chromosomal abnormality, chances are it will undergo a spontaneous abortion. Right? And approximately 1 out of every 155 live-born children have a major chromosomal abnormality. Most of those are Down syndrome. Right? Again, so that's the major, that's the most probable trisomy to have and to live with. So about 13 individuals of every 10,000 live births have trisomy 21. Right, the 47 chromosomes. We said Klinefelter syndrome and Turner syndrome are also more common. You have XYY, Jacob syndrome, triplo X, trisomy 13, trisomy 18. So altogether, about 33 individuals between 10,000 live births can actually survive with an extra or missing chromosome.